Endoplasmic reticulum. General history of endoplasmic reticulum. Early cytologists held that some sort of supporting network or cytoskeleton was present in the cells. It was given various names such as nasal substance, ergastoplasm, basophilic bodies, etc. In 1945, Porter, Claude, and Fullman, with the help of electron microscope, noted a delicate membranous network in the cytoplasm. It was later called endoplasmic reticulum by Keith Porter in 1953. The endoplasmic reticulum originally seemed to be confined to the endoplasm of the cell, hence its name. Structure of endoplasmic reticulum. It represents 30 to 60% of total membrane in a cell. Endoplasmic reticulum comprises of three types of elements named cisternae, tubules, and vesicles. Cisternae. These are flattened, unbranched, sac-like elements which are around 40 to 50 micrometer in diameter. They lie in stacks parallel to but interconnected with one another. They are separated from one another by cytosolic spaces. The small granular structures called the ribosomes may or may not be present on the surface of cisternae. Vesicles. These are oval, vacuole-like elements about 25 to 500 micrometer in diameter. They often occur isolated in the cytoplasmic matrix. They are also free of ribosomes. A fluid called endoplasmic matrix is present in the lumen of endoplasmic reticulum. All the elements of endoplasmic reticulum freely communicate with one another. Tubules. These are irregular branching elements which form a network along with other elements. They are about 50 to 100 micrometer in diameter and are often free of ribosomes. Ultrastructure of endoplasmic reticulum. The membrane bonding the cisternae, tubules, and vacuoles of endoplasmic reticulum is similar to the cell membrane. It is 50 to 60 angstrom thick. The membranes of endoplasmic reticulum are composed of two layers of phospholipid molecules sandwiched by two layers of protein molecules like other membranes in the cell. The endoplasmic reticulum membrane has relatively high protein-lipid ratio. It is continuous with the cell membrane, Golgi membrane, and outer membranes of nuclear envelope. Certain cisternae open out by pores in the cell membrane. In the lumen of endoplasmic reticulum, secretory granules were observed by Pallad in 1956. The lumen acts as a passage for secretory products. About 30 to 40 different enzymes are associated with endoplasmic reticulum for various synthetic activities. These may be located on the cytoplasmic surface or luminal surface or both. Membrane-bound endoplasmic reticulum spaces varies in shape and size in different cell types. On the basis of presence or absence of ribosomes, there are two different kinds of endoplasmic reticulum found in the cells. Smooth endoplasmic reticulum. Ribosomes are absent on the walls of endoplasmic reticulum, and so it appears smooth and hence called smooth or agranular endoplasmic reticulum. It mainly occurs as smooth forms, and the tubule forms irregular lattices and measures about 500 to 1000 angstrom in diameter. Smooth endoplasmic reticulum is commonly found in the cells involved in the synthesis of steroids or lipids. Certain cells with carbohydrate metabolism, with pigment production and electrolyte excretion. Functions of smooth endoplasmic reticulum. It provides surface for synthesis, glycogen metabolism, detoxification, formation of organelles, transport route, skeletal muscle contraction, fat oxidation. Rough endoplasmic reticulum. It is characterized by the presence of ribosomes on the surface of reticulum, and so it is also known as granular endoplasmic reticulum. It is in the form of flattened cisternae with the width of 400 to 500 angstrom. Rough endoplasmic reticulum occurs largely in the cells that are actively involved in the synthesis of proteins such as enzymes or mucus. In exocrine cells of pancreas, rough endoplasmic reticulum consists of reticular sheets and fenestrated cisternae in the basal region of the cell. These cisternae measures about 5 to 10 micron in length, and their groups are 400 to 1000 angstrom in diameter. In apical region of cells, the granular reticulum occurs in the form of vesicles. Granular and agranular endoplasmic reticulum are in continuity of their membranes in the regions of contact. Functions of rough endoplasmic reticulum. Surface for ribosomes, surface for synthesis, packaging, smooth endoplasmic reticulum formation, formation of nuclei, formation of glycoproteins. Thank you.